I'll be talking today about the evidence for TAAR1 as a potential therapeutic target in psychiatric disorders. In patients with schizophrenia, there are multiple clusters of symptoms, such as positive symptoms, including delusions, hallucinations, disorganized thinking, negative symptoms, including blunted affect, avolition, emotional withdrawal, asociality, anhedonia, and alogia, as well as cognitive symptoms. In addition, there are motor symptoms that preceded the onset of antipsychotics. So multiple domains of function in the brain is affected in patients with schizophrenia. One medication, one therapeutic target that's been the kind of the main target so far has been the D2 receptor, with antipsychotics being utilized to treat patients with schizophrenia. These are referred to as first-generation antipsychotics and second-generation antipsychotics, but they essentially do not differ much in the mechanism of action, as they all have in common the fact that they interact with the D2 receptor, but essentially by blocking it, antagonism of the D2 receptor. Some of them have additional effects at other therapeutic putative targets, such as the 5-HT2A receptor. And some of them may interact with the D2 in a way that's different from full antagonism. So, for example, partial agonism. They mostly differ in their side effect profile. Some of them induce weight gain, and some produce more motor symptoms, hyperprolactinemia, etc. So this treatment is mostly based on the fact that there are dopaminergic abnormalities in patients with schizophrenia. This has been a long-standing hypothesis in the field. It's based on the concept that there are neurodevelopmental and genetic factors that affect the neural circuitry between different dopaminergic brain areas. And the idea was initially that there is an excess in dopamine in the striatum, especially in mesolimbic pathways that produce psychosis, and a deficit in dopamine in the cortex that produces cognitive symptoms and contributes to negative symptoms. As I have mentioned, antagonism at D2 receptor is the main target of all antipsychotic drugs that we have available at this point. But in addition to dopamine, there are also other neurotransmitter abnormalities in schizophrenia, such as those with glutamate, the main excitatory neurotransmitter in the brain, GABA, the main inhibitory neurotransmitter, and 5-HT, which modulates different other neurotransmitter systems, including dopamine. In terms of dopaminergic pathways in the brain, there are 4 that are relevant for our discussion of schizophrenia. The mesocortical pathway projects from the ventral tegmental area to the prefrontal cortex. It controls cognitive functions, including attention, executive function, planning, and abstract reasoning. Hypodopaminergic activation is thought to underlie both the negative and cognitive symptoms of schizophrenia. The hypothalamo-hypophyseal tract, and this is a dopaminergic projection from the hypothalamus to the anterior lobe of the pituitary. Dopamine in this pathway chronically inhibits prolactin release. Antipsychotics antagonize dopamine receptors, D2 receptors, and this way they release the inhibition of dopamine onto prolactin, which produces the elevation in prolactin that we see in patients with schizophrenia. The nigrostriatal pathway, this one projects from the substantia nigra to the striatum. It's involved in motor control, but also in cognitive processing. D2 antagonists decrease dopaminergic activity in this projection field, causing EPS. Evidence suggests that 2A antagonists may increase dopamine release in the striatum, which might result in lower incidence of EPS with second-generation antipsychotics. Finally, the mesolimbic tract. This pathway projects from the ventral tegmental area to the limbic structures of the nucleus accumbens. This pathway is involved with motivation, reward, long-term memory, and emotion. Hyperdopaminergia in this pathway is thought to be involved in the positive symptoms of schizophrenia. Now, the imaging studies have been used, positron emission tomography, multimodal imaging, to examine this dopaminergic hypothesis in schizophrenia with the striatal excess cortical deficit, mesolimbic hypothesis, and these imaging studies have contributed the following information. The mesocortical deficit was confirmed using that imaging. Within the striatum, what was observed is that there is an increased dopamine release and synthesis, mostly in the associative striatum, which is the territory that receives projection from the nigrostriatal dopaminergic projection to the midsection of the striatum, and that is involved mostly in cognitive processing. This excess dopamine in the associative striatum was shown to relate to more psychosis, better response to antipsychotics in those patients, weaker connectivity of the rostral caudate, part of the associative striatum, to the rest of the brain and the cortex in particular, and more perceptual bias predisposing patients to more hallucination. So excess dopamine in the striatum increases the perceptual bias to be influenced by what patients are expecting to hear rather than what the stimulus actually is. And that is the primary cognitive mechanism for hallucination. So these were all observed in vivo, giving a lot of supportive evidence for the dopamine imbalance in schizophrenia. To summarize, dopamine release is decreased in mesocortical pathway. Dopamine release is increased, and synthesis, I should say, is increased in the nigrostriatal pathway, especially the one projecting to the midsection of the striatum. So now this brings us to the trace amine-associated receptors. So these are G protein coupled receptors that are physiologically activated by low level of trace endogenous biogenic amines. There are multiple biogenic amines in the brain at very low concentrations. TAAR1 is the most studied TAAR and is expressed in both central and peripheral tissues in the brain. It's localized to structures associated with psychiatric disorders. For example, it's found in the VTA, the dopaminergic region that's associated with learning, motivation, reward, salience. And the dorsal raphe nucleus, which is a serotonergic region that is involved in multiple aspects of behavior and cognition. And the prefrontal cortex, obviously a region very involved in cognitive functioning, impulsivity, and which a region rich in glutamatergic and GABAergic circuitry. Peripherally, TAAR1 is localized in the gut and tissues associated with energy metabolism. So TAAR1 is a therapeutic target under investigation in patients with schizophrenia because TAAR1 can act as a modulator regulating dopamine, glutamate, and serotonin activity. And we have evidence involving all of these transmitters and the pathology of the brain in patients with schizophrenia. Some more than others. For example, I've discussed the evidence supporting dopamine. MRS studies have incriminated glutamate and GABA in the cortex in patients with schizophrenia. And there's less support for serotonergic abnormalities in schizophrenia, but the addition of the 2A antagonism to drugs seems to lead to needing lower D2 occupancy for therapeutic success, which indirectly incriminates the serotonergic system. So dopamine, which obviously is a main target of treatment in schizophrenia, can be modulated by TAAR1. It can be downregulated by activation of TAAR1, and it can be enhanced by inhibition of TAAR1. So obviously, in light of the excess in the striatum, activation of TAAR1 would be a logical therapeutic potential. TAAR1 compounds have demonstrated activity in a wide range of animal models suggestive of antipsychotic, antidepressant, procognitive, anticatalyptic effects, all important areas for schizophrenia. And we'll look at some of this evidence in later slides. There are also genetic studies suggesting a link between TAAR and schizophrenia. For example, the TAAR1 gene's locus overlaps with other susceptibility loci. Some key enzymes involved in the synthesis and degradation of trace amines have been associated with schizophrenia, and TAAR1 rare variants have also been detected in patients. Two TAAR1 agonists, relmitaron and ulutaron, are currently in clinical development for the treatment of schizophrenia. So the evidence suggesting that TAAR1 agonism may be useful on the positive symptoms of schizophrenia comes from studies in preclinical models and animal studies showing, for example, that TAAR1 knockout mice are hypersensitive to amphetamine-induced hyperlocomotion, hence striatal dopamine release as well as serotonin, norepinephrine. So basically, if TAAR1 is absent, as in the knockout, there is no protection anymore against this excess surge of dopamine. TAAR1 agonists dose-dependently can block cocaine-induced hyperlocomotion, similar to the antipsychotic olanzapine. So again, counteracting the excess from the cocaine-induced surge in dopamine. They can potentiate the effects of suboptimal doses of olanzapine for blocking cocaine-induced hyperlocomotion, and they can block PCP-induced hyperlocomotion. So this could be an NMDA receptor-mediated effect, as well as potentially an effect on dopamine from the NMDA interaction with dopamine. In terms of evidence for TAAR1 effects relevant to cognitive and negative symptoms of schizophrenia related to the mesocortical tract and possibly to the mesolimbic tract, TAAR1 agonist administration decreases VTA activity in the intracranial self-stimulation test. So obviously they are active within the VTA on dopamine cells. As measured by pharmacological MRI, TAAR1 agonists increase perfusion in corticolimbic areas, suggesting enhanced prefrontal cortical activity. TAAR1 agonists improve the accuracy and object retrieval test in non-human primates, so direct effect on cognition. And TAAR1 agonists improve attentional set shifting, deficits produced by subchronic PCP treatment. So again, counteracting NMDA dysfunction and glutamate dysfunction. In terms of TAAR1's effect on extrapyramidal symptoms produced by antipsychotics, so relating to the excess blockade in the nigrostriatal pathway, haloperidol significantly increases cataleptic time in rodents. That is a model for extrapyramidal symptoms. TAAR1 agonists do not produce these type of symptoms in animal models, so no muscle rigidity or other signs of catalepsy. And TAAR1 agonists partially attenuate the Haldol-induced catalepsy. In terms of potential effects of TAAR1 on metabolic syndrome, prolactin, or other effects from the hypothalamic hypophyseal tract, what we know is that olanzapine increases body weight and fat mass in rodents. These are attenuated by TAAR1 agonists. In addition, TAAR1 agonist effects on body weight and fat mass are similar to placebo. Effects on neurotransmitters. As we've mentioned earlier, TAAR1 agonism counteract dopamine, modulates dopamine activity. So when we increase TAAR1 activity with an agonist, it lowers dopamine neurotransmission, and vice versa. Lowering TAAR1 activity increases dopamine transmission. For serotonin, TAAR1 is expressed in the DRN, dorsal raphe nucleus, and agonists at TAAR1 decrease the firing frequency of serotonergic neurons in the dorsal raphe nucleus of mice, suggesting modulation of serotonergic system. TAAR1 likely functions endogenously to maintain a balance in serotonergic activity. In addition, TAAR1 interacts with the glutamate system, can regulate glutamate neurotransmission in the prefrontal cortex. TAAR1 appears to prevent hypoglutamaturgic states, thus improving function in the prefrontal cortex. So in summary, schizophrenia is a highly complex disorder associated with both genetic and neurodevelopmental risk factors. Dysfunction of the dopaminergic system has been well documented, as well as dysfunction in glutamatergic, GABAergic, serotonergic systems. Currently approved pharmacological therapies for the treatment of schizophrenia focus on the D2 receptor, mostly antagonism or functional antagonism. TAAR1 is being researched as a potential novel therapeutic target for the treatment of schizophrenia. It's a protein-coupled receptor that modulates presynaptic dopaminergic neurotransmission in preclinical models, modulates dopamine, glutamate, two additional neurotransmitters that are important to the pathophysiology of schizophrenia. Thank you for your attention.